for inviting me here. And when I contacted him, I didn't actually ask to give a seminar. I said the only reason I wanted to come up here is I wanted to meet him. And the reason is because he didn't tell you, I never met Phil's father. He was one of those rare people who was kind and sent tissues to us at a time when we weren't even known in the field of epigenetics. In fact, and I'll get into this, it's because of him that we were able to get into the field of epigenetics in a very high visibility way. And this is a lesson maybe to all of you, that sometimes kindness to people that you don't even know can have tremendous effects, not only in the person, but really on the whole field. So I never met Barry, nor did I meet Phil's mother, but I wanted to meet him. And so that's the reason I'm here. And we just met literally yesterday in the airport, 18 years after your dad sent us these tissues of the echidna and the platypus, which you'll see was absolutely critical for our research at that time. So I start out these talks, and this is kind of a different talk than I normally give, so I was telling Phil it might go a little bit longer, but I'll try to get it going fast. I told him I can play, as my wife can tell, I can talk forever. So if there's people need to go, and I do appreciate your coming here, give me the high sign and we'll wrap her up. So today I'm going to talk about epigenetics and how the genes and environment interact. And we know, because we all read the papers and newspapers and scientific literature, we're still really in the era of the genome, even though I've been doing work in the field of epigenetics since the early 90s. The vast majority of your work is still looking for genetic mutations that are being trying to be linked to various diseases and neurological disorders. And this has been successful, obviously, and I think because I came through a cancer background. BRCA1 and BRCA2 mutations, where 80% of the women that have this are going to have trouble either with breast cancer and or ovarian cancer. And that's a high penetrance of mutations that occur in these genes. But many of the disorders, like the chronic ones, like diabetes, obesity, neurological disorders, and you think of autism and schizophrenia, this approach has not been as successful. They can account for some small variants, and then they add them all up and it gets a little bit bigger. But they're still missing 60 to 70% of the variants. So where is that? So to me, a more interesting question really is, why do identical twins also vary in their disease susceptibility if the only thing that's important in this whole process is genetic variation? And this is showed very nicely by this painting that I had commissioned by Colin Murphy, who, by the way, was on the faculty in science at the University of California, San Francisco. So not only is she a great scientist, she's a great artist, and she's a pianist, so she's art also. I like her paintings because they're bright, vivid colors. And it's hard to get this concept across on a painting, that the environments can alter. So even though they have the same genomes, they more than likely don't have the same epigenomes. And the reason for this is that they're not necessarily, and most likely are not, in the same environments, even though they're in the same womb. And the reason that that can happen, one reason is, for example, they have different blood flows. So I show this here, the different environments are just different washes over these developing embryos. And as a consequence, the epigenomes can be completely different and therefore give rise to a very different characteristics. I mean, people can usually tell identical twins from each other, usually behavioral, but even sometimes facial features and stuff are not the same. So those of you that don't know what epigenetics means, and it's probably most people do, but I'm just going to go through this very briefly. I'll define it. It's a very easy definition. Epi just means above, like epicenter is above the center. This is just above the genetics. But research refers to it this way, the formal thing, is a study of heritable changes in gene function that occur without a change in the sequence of DNA. And the two main components of this epigenetic code is one that's physically on the DNA, this is the main one anyway, where methyl groups are attached to cytosines when they're 5' of guanine, and these are involved in regulation of genes. And usually when you have these methylation groups attached, it causes the gene to not be expressed. And that's coupled with basically changes that are also epigenetic changes that are present on these little tails of the histones that the DNA is wrapped around. And in combination, they cause either the DNA to be compacted and as a consequence not functional, or opened up and therefore functional. So I like to think about the genome as being comparable to this physical hardware of a computer. And then the epigenome is this basically the software that tells the computer when, where, and how to work. So this is how you can have the same genome in every cell type, but yet you have 260 different cell types. In effect, it's like taking 260, I like Mac, so 260 Mac computers and line them up. And every one of those computers is running a different program. And that program in our body basically is established during early development. And so now you have a mechanism by which you can tell from the same genome, you can create different cell types. So I come from a background of physics and math. I got my undergraduate degree in nuclear engineering and computer science. So I think of a cell as being a programmable computer. If you think of it this way, it's easier to understand what's going on here. Now, if there are any questions, please ask them, because this is not a big group. So if it's like a burning thing that I just haven't explained or you don't think I'm going to explain, just raise your hand and I'll try to address it at the time. So for me, the field of epigenetics began with this person, Denise Barlow, who unfortunately died in October of 2017. A phenomenal scientist. And she had a very different, and I think kind of neat definition of epigenetics, and this was her definition. Epigenetics has always been all the weird and wonderful things that can't be explained by genetics. It's a great definition. So what did Denise do that got me literally into the field of epigenetics? This is what she did. She identified the very first genomically imprinted gene, which in mice, which is what she was working in, was the IGF-2 receptor. So imprinted genes, for those of you, like if you're working all in aquatic animals and stuff, you'll never run into these genes, and I'll tell you why later on. But in mammals, these are very, not common, but they're definitely a number of them in our genomes. So imprinted genes are autosomal genes with a sex. And you have to ask the question, how can it be? What's the machinery within the cell that allows for either the maternal copy or the paternal copy expressed and only one copy expressed, depending upon the gene? The reason is because the marks are placed differently in the egg that control this than there is in the sperm. So I'm going to go through this briefly. All this work was done by Denise Barlow. Amazing stuff. So how did this gene bring me into the field of epigenetics? Right after she discovered this to be an imprinted gene and therefore was expressed only from the mother's copy, the father's copy was silenced epigenetically. We define this gene as being a tumor suppressor gene in basically every cancer that's been looked at. This is a tumor suppressor. So I'm thinking, this is astounding. We've got two copies that we inherit, and on purpose, Mother Nature has silenced one of those copies, making us functionally haploid at this locus. And there's about 500 of these genes at least sitting in our genome, all involved to a great degree in growth regulation, and therefore probably all of them are somehow involved in cancer formation. Either too much of it, which would be called an oncogene, or too little of it, which in this case is a tumor suppressor gene. So the mechanism by which this occurs is very simple. I'm going to go up in a minute because this is just really great. There's a transcript that comes off. This is the very first. This thing is 100,000 bases long. It was so long that it probably destroyed the career of a couple of postdocs. Because this is the good old bad days when no sequence was known. And they kept sequencing and sequencing and sequencing, and it kept getting bigger and bigger and bigger. And finally, they hit the end at the 100,000 bases. Big, long RNA transcript. So this is only expressed, this is incredible. It's only expressed from the father's copy because this differentially methylated region right here. Now, differentially methylation is often meant between one individual and another individual. This is not what I'm talking about. I'm talking about the maternal versus the paternal copy of this gene, okay? So the maternal copy is methylated right here. This is DMR2, it's referred to as. And as a consequence, this promoter region is shut down and there's no antisense made, okay? Whereas in the father, in other words, coming in from the sperm, this area is not methylated. And as a consequence, the transcript is made, and once it runs through the promoter region, it brings in the machinery that methylates yet another DMR, which is right here, DMR1. So it's methylated here and absolutely requires the RNA species to be produced, because when Denise Barlow truncated it, this does not become methylated. So it has to run through this region. It's now methylated. So the promoter region now for the IGF2R is now shut down, and there's no expression of the gene off the father's copy. Whereas the mother's copy, this DMR is never methylated because the antisense doesn't go through, and the only copy that is expressed is the one from the mother. So usually you think of DNA methylation as being required to shut down something. In this case, the inherited part of this, the one that comes in the egg and the sperm, that region, the methylation is required to keep the maternal copy open. Right? So this is a differential methylation that you get at this gene. These DMRs are present for every imprinted gene in our genome. They are not known except for about 20 or so genes, and right now at State, we are in the process of trying to determine these DMRs throughout the whole genome, so we have what is called the human imprintome, another ohm, of all of these regions. Once we know this, we can screen any disease, environmental state, disease state, for which genes and which DMRs are deregulated. But we can't do it now because we don't know the human imprintome. So as I said, when you first heard this, and you know that you've got tumor suppressor genes that are being inactivated, and then IGF2, which was also being shown later on to be imprinted, that's an oncogene, which now you get too much expression because of imprint deregulation. You have to ask the question, why would something this nutty ever evolve? And David Haig asked that question and came up with a theory. Now David Haig, which this time I actually put his name here so I wouldn't forget to mention him, is a native born and raised in Australia. And I think he's actually going to be doing a visiting professorship, I believe, down at University of Adelaide. And you might want to get him up here because he's a great speaker. He then postulated in 1991, after the IGF2 receptor was identified as being the first imprinted gene, that if IGF2, which is the growth factor, was imprinted, that it would be expressed from the father's copy. The mother would shut down her copy and it would express only from the father's copy rather than the mother's copy. And the theory then suggests that right now there is really no advantage of these to us personally, but they are basically a genetic remnant, or a remnant of a genetic battle between males and females to control the amount of nutrition that the offspring extracts from the mother, with the father trying to maximize that extraction for propagating genes forward, and the mother trying to dampen it down. So you've got this potential genetic yin and yang at these two different loci. And indeed, the next year IGF2 was identified as being imprinted and expressed from the father's copy, just like he predicted from this theory. Now that doesn't prove it, but it's consistent with it. And many of the imprinted genes now fall in this sort of thing. Paternally expressed tend to be pro-growth. Maternally expressed imprinted genes tend to dampen down growth. If you're interested, though I won't talk about it, this battle, as you would expect, will show occurs in the placenta, and the other place where imprinted genes are heavily expressed is the brain, and probably involved in neurological disorders. And Christopher Babcock from London has now come up with an extension of this theory that goes along how these genes are involved in autism and schizophrenia. If you're interested, look up his name. So it made one other important prediction, and that is that imprinting then would be expected to not be present in animals that lay hard shell eggs, because the father could not control the amount of nutrition that the mother puts into those eggs. That was a theory or a prediction that we could test. And it would enable us to get into the field of epigenetics relatively big time quickly, because we were not known in the field at all. But in order to do this, we had to have tissues from the monotremes. Because there's now 3 living groups of mammals on Earth, monotremes, of which there's 2 members basically of that. There's the echidna and the platypus. Then there are the marsupials, which in North America we do have the opossum, though most of them are down here in Australia. And then the eutherians, which basically is the vast majority of mammals on Earth. Now, you guys probably know this, but I like asking it. Does anybody know the definition of monotreme? Anybody? No? It means one whole. Because the urinary tract, gastrointestinal tract, and reproductive tract exit from a common orifice called the cloaca. This is a mammal that lays eggs. Now, when I asked our students at Duke, we have really smart students at Duke University, I asked one, nobody knew that. Finally, somebody raises their hand way in the back with vigor. And I said, yeah, what? He said, it means one treme. He was correct, but it means a little bit more than that. So the problem with this then, and our ability to do this, was getting tissue. And this was where Phil's father came to the rescue. He sent us platypus tissue on dry ice, and as a consequence then we could look at the evolution of genomic imprinting. And when we did this, we had some very interesting results. So these are the 3 papers. Keith Killian was an MD PhD student that did this work in the laboratory, and these are the 3 papers that we co-authored with Barry Monday. So when we did this, the first thing we saw, both at the IGF-2 and IGF-2R receptor, is that if you look up and down the phylogenetic tree, birds did not have imprinted genes, at least at these 2 loci. Monotremes did not, whereas marsupials and eutherians did. So that it showed for the first time that at least at these 2 loci, that imprinting evolved about 150 million years ago in a common ancestor that gave rise to marsupials and eutherians, which collectively are called therian mammals. Whereas monotremes, which lay eggs, birds, and then we even looked at skinks and stuff like this, there's no evidence of genomic imprinting. Fish would not have them, etc., in any of the species that are more ancestral. So if you look up and down the phylogenetic tree for IGF-2, every species that's been looked at, it's good evidence that the paternal allele is only expressed and the mother's copy is shut down. The IGF-2R is really a lot more complicated. Now I have to tell you, the IGF-2 receptor is not the signaling receptor for IGF-2. The signaling receptor for IGF-2 is the IGF-1 receptor or the insulin receptor. The IGF-2 receptor is the degradation pathway for IGF-2. So when you knock this out in cancer, what you have basically is a lot more extracellular insulin-like growth factor, which is pro-carcinogenic. Okay? So that's why it functions as a tumor suppressor. It's not a signaling receptor, it's a degradation pathway. It binds these compounds with, anyway, it's got a number of binding sites, but it binds IGF-2 and internalizes into the lysosomes for degradation. So if you look at IGF-2R, it evolved imprinting here before marsupials, but when we looked at it as far as degradation, I mean as far as in primates, we found no evidence of IGF-2R imprinting. There was also not in tree shears and the flying lemur, which doesn't fly, and it's not a lemur, it's also not imprinted. So IGF-2R imprinting was actually lost 75 million years ago. So what this showed for the first time, that imprinting not only can evolve, but you can also lose it if there's pressure to do so. On top of that, even though those physical marks are there that imprint to have monoallelic expression, it's like this. Just because I write this piece of paper that marks those genes for imprinting, it doesn't mean that you have to read them, right? And it's the same thing with the imprint marks. So as a consequence, monoallelic expression in a parent of origin dependent manner is species dependent. We showed that here, but also tissue, time, and sex dependent. So the marking is species dependent, but the reading within the species can vary greatly dependent upon the tissue, time, and sex of the animal. You can imagine if you would be imprinting, which there's now evidence in humans, Hox genes, which are changing physically the patterning, the body plan of animals, you could potentially have an animal that walks on 4 legs stand up and walk with this type of phenomena going on. So it appears that once imprinting was evolved, that it was used to speciate. But it would only be happening, obviously, in theory in mammals, because they're the only animals on Earth that we know of anyway that have this type of regulation of gene expression. I'm going to end this now with one more slide, and this is just an interesting slide from my standpoint. So here you got this beautiful painting, the Jurassic area where the dinosaurs ruled. And don't those trees look like the ones we were talking about? They were nibbling on these trees a long, long time ago. So back at about 60 to 65 to 70 million years ago, when the asteroid hit the Yucatan Peninsula and you had the KT boundary, which eliminated 85% of the species on Earth, these dinosaurs basically disappeared. But you notice in this picture, there are 2 other little animal things live. Mammals were here still back in that time, and angiosperms were present. Right? They were there even then, but they didn't play a big role, but they were there. After the asteroid hit, the angiosperms and the mammals basically filled up all the niches. And they're the only things on Earth that we know of that have imprinted genes. Now, does that mean anything? I don't know, but it's an interesting association. So, once we did this, we knew that we wanted to determine whether the environment, and I didn't know what, just environment in general, could potentially alter the epigenetic regulation of these, what I call epigenetically labile genes, of which one group of them would be imprinted genes, and the other group is called metastable epialleles. And that I'll talk about in greater detail now. That terminology, epigenetic epialleles, was coined by another Australian scientist, Emma Whitelaw, who is now, I think, retired. Another fantastic scientist in the field of epigenetics. So we're going to start this part of the discussion with the developmental origins of health and disease, because our studies in this field of looking at whether the environment can alter the epigenetic regulation of genes brought us directly right into the heart of this field. It's also called the fetal origins of adult disease susceptibility, and it's also referred to as the Barker hypothesis, so because he's the one that did the original work in this area, working with the Dutch famine survivors. So this, in 1944, 1945, that winter was particularly harsh in many respects, weather-wise, but also the Nazis put an embargo on Holland, the part that they still controlled, limiting the amount of food that could get into that area severely, so that the average person was eating somewhere around 700 calories a day. As you know, normally it's 2,000 to 2,200 calories. So this is a severe restriction in food. 16,000 at least people died during that famine in Holland, but there were women that got pregnant. And interestingly, some of these children were born. Now they were small size at term, but they were born and they survived. And Barker and a number of epidemiologists followed these people, and lo and behold, they found that 20 and 30 years later, that the children that were in utero, particularly during the first trimester of this starvation diet, had high incidence of cardiovascular disease, obesity, and interestingly, a doubling of the incidence of schizophrenia. Unfortunately, this natural experiment was replicated in the late 50s and early 60s in China, and again, 20 to 40 million people died during this famine. And again, the offspring were shown to have, in effect, the same kinds of characteristics that were shown in the people that were exposed to the Dutch famine, increases in obesity, diabetes, and schizophrenia, these chronic disorders that we hear about and have problems with now. But the state of the art of this field, I think, was nicely demonstrated by one of my favorite cartoons, Then a Miracle Occurs. So what you have here is a student trying to tell his professor that he had proved this wonderful mathematical proof where he starts here and he ends here, but in the middle we don't quite know what the hell's going on, right? Yeah, have you ever heard that? So this is by Sidney Harris. So in effect, we have the same thing here. We know that severe lack of food early in development, the earliest stages of development, is linked to increases in the number of chronic diseases 20 and 30 years later. But the problem was there's a miracle that occurred. What's the gravity, the glue, the memory system that connects these two incredibly disparate time points? And we showed in 2003, at least in animals, and now has been also demonstrated, at least from association studies, to occur in fact, the Dutch famine people, that indeed that miracle involves epigenetic modifications. Not a miracle. You just didn't know what it was, right? So here's the study we did. So for every problem in a given discipline, I love this. This is E.O. Wilson, who is a behavioral biologist at Harvard. He wrote, for every problem in a given discipline of science, there exists a species that's ideal for a solution. You just got to find it, right? So from the standpoint of looking at environmental epigenomics, which is what that study that we did ushered in, the era of the environmental epigenomics studies, the ideal animal are the Agouti mice. I call these the Agouti sisters. These are offspring. They're both 100 days old. They're both female. And they're genetically identical. So if you look at that, knowing what I just told you, and don't appreciate the importance of epigenetics in disease susceptibility, I hate to say it, but there really isn't any hope for you. None. There isn't a clear example of this. Now, we didn't develop this system, but we really used it well. And that's the importance about these model systems. You've got to find the appropriate ones that have the least problems and use them well and effectively. So how does this work? Here's your transposable element. Tim, you probably know all this already, but I'm going to go over it. So the first thing that happened is the crazy little strain of C57s. This is on the C57 background. A little transposable element jumped in upstream of the Agouti gene. This is a mutation, right? It's a mutation. Popped in here. What it did then in this situation, it created an alternative start site for the control of the Agouti locus. So normally the Agouti gene is regulated developmentally. So you have a black hair shaft, right? And right at the end of the development of the hair follicle, the hair shaft, it turns on the gene and it puts a little band of yellow right at the base of a black hair shaft. We now see the mouse not as black, but as Agouti. It's brown. So if any of you work with C57 mice, have you ever seen those? All of you have never seen these mice. If you did see them, they're completely black. And the reason for that is this Agouti gene is mutated. So even though it's turned on, it doesn't work. So you just have a complete black hair shaft. Now in this strain of animals, you got a very interesting little system here. So if this region here, this transposable element, very early in development is methylated and appropriate histone marks are put on it, it shuts the thing down so that it can't, as you were saying with temperature, it can't jump around in other places. It shut it down. So this thing is shut down. As a consequence, it's regulated developmentally, and you get a brown mouse, right? Just like you would normally expect. Whereas the decision is made to not methylate this region, so there's no methyl groups here, or very few, then it turns on the Agouti gene inappropriately throughout the whole animal's body, including the brain. And in the brain, the protein, the Agouti protein, binds to the melanocortin-4 receptor, so it blocks the satiation center of the animal, and as a consequence, it never knows it's full, and it literally eats itself into obesity, diabetes, and cancer. So who would have ever thought that inappropriate expression of a Agouti gene would be involved in obesity, diabetes, and cancer? But in this model system, in fact, it is. So if you have a yellow animal, or even one with just a little bit of yellow on it, you can guarantee this is what's going to happen in this model. So anything that potentially skews methylation and increases the probability of this region being methylated is good, and anything that reduces it will skew it towards this side and is bad, okay? Good is brown, bad is yellow in this model system. And that good and bad is regulated by methylation of this transposable element that just randomly went in here. So in effect, what you're doing is counteracting the effect of a mutation by epigenetic phenomena. It's amazing. These are the modifiers. So you ask the question, I don't know if this is true, so don't go home and tell everybody, but when BRCA1 and BRCA2 mutations occur, 80% of women get cancer, but 20% don't. Is it possible that something like this potentially is going on in these women so that they're counteracting the negative effects of the mutation? I don't know, but I think maybe we should probably look at this. Because if it is, there's potential ways in which you could ramp this up maybe with the use of compounds and protect against these mutations. Now, please don't go home and say this is correct because I don't know. But here we do know that that kind of thing can happen. What you're doing here is you're negating the negative effects of this mutation on this gene by methylating it, right? So Rob Waterland, who was a nutritionist, came into the laboratory and wanted to look at something as simple as nutrition. Does this potentially affect the regulation of these genes epigenetically? And we chose this model system to do these types of studies. So if you look with control diet, this is just normal mouse diet that you buy, you find that the vast majority of the animals, offspring, are yellow or nearly yellow, right here in this area. And there's about 10% that are blonde and about 10 to 15% that are brown. But the majority of the animals are over here. So it's a normal distribution. It's maybe a little bit skewed towards yellow. So then we put the mother's diet had methyl donors, which is choline, vitamin B12, folic acid, and betaine. And 2 weeks before we put the mother on the diet, all the way through gestation and up to the time they were weaned, which is 20 days afterwards, then we took the samples at that point. When he did this and looked at the coat colors of the offspring, this is what he found. Indeed, when you increase the amount of methyl groups, because all the methyl groups come in from your diet, from these compounds, you greatly increase the incidence of animals that were brown or nearly brown. That was exciting, but what was most important was this. When he looked at the methylation in this area of the IAP, there's 7 sites that he looked at, cells that are methylated versus site. Control diet, the vast majority of the animals are hypomethylated. There's very little methylation. There's some up here, but very few, which correlates absolutely with the coat color of the animals. Then when you supplemented the mother's diet with these methyl-doning supplements, you can clearly see that the level of methylation that occurs in these animals was greatly increased. As I said, even the untrained eye without statistics can see that this has significantly increased the methylation in addition to coat color, which are now brown or heavily brown. So that for the first time we demonstrated that something as subtle as maternal nutrition can affect disease susceptibility and coat color in this model system by alterations in the epigenome. This ushered in the era of environmental epigenomics, which you guys are now looking at yourself, because another stressor on this system can be temperature, CO2, any kind of environmental change potentially can change the ability to modify these methyl groups and where they're put. So as I said, we had the control diet. Then what we did is we asked the question, there was evidence that genistein, which is present in soya, and it's high, women that are in Asia have high levels of this, and it appeared that it was potentially reducing breast cancer incidence. We looked at this is work of Dana Dolanoy, who's now at the University of Michigan. And indeed, what was interesting, she also found that genistein at levels present in women in Asia also increased the incidence of brown animals, which in this situation is a positive adaptive response and protective. Now, what was so important is not only that, is the fact that genistein doesn't have a methyl group to donate. So other compounds that don't do things like we were first studied can also affect the laying down of these methyl groups in this model system, the protection of these animals from these negative effects that you see when that transposable element is not methylated. That was the first evidence that indeed that could happen. Then the next thing Dana did, because she came from a tox group, wanted to know whether the estrogenic compound bisphenol A, which is present, used to be anyway, in water bottles, baby bottles in the United States. I think it's still in cans. If you open up cans, you have bisphenol A as a layer that's inside of them. Teeth, the coatings on children's teeth, bisphenol A. We're all exposed to lots of bisphenol A. And at least in rodent models, this compound causes cancer formation. It's not a good compound, at least at higher levels. And indeed, when she put the mothers on a level of bisphenol A that's comparable, it's an endocrine disrupting agent, that you see in animal models and even in humans, what we see is an increased incidence of the yellow animals, which in this model system is bad. So if you look at this curve and this curve, it's not hard to think, is it possible, since this is plastic, you can move it back and forth, that you might be able to negate the negative effects of bisphenol A either by supplementing the mother's diet with methyl donors or genistein. And when we did that, lo and behold, we went right back to control. So the important thing of this study was not really that bisphenol A altered the epigenome, which is important, but the fact is that nutritional supplements can modulate this in this situation in a good way. So I was so excited about this. We submitted it to PNS and it was accepted, that I had asked Colin Murphy if she would do a painting for us that would sort of show this. So here you've got water bottles dumping bisphenol A down on developing embryos. This is a great painting. Great, just bathing in badness, right? And to its rescue, sort of like Superman or woman, comes the vegetables to counteract the negative effect of bisphenol A that's being dumped on you through these plastic bottles. This was such a damn great painting that PNS decided not to put it on. It's still a good painting, though. It's really great. So, as Harpocrates said 2 millennia ago, let food be thy medicine and medicine be thy food. Indeed, at least for diseases that are caused through epigenetic deregulation, food is medicine. You got to remember this. It is important. The clinical people are there to treat things, but it's possible that we can prevent some of this stuff by our actions, what we do, what we eat, etc. That's what these results really, I think, clearly show. Now, I told you that my background was engineering. I got my undergraduate degree in nuclear engineering and computer science. So the last study that I did at Duke before I retired, I wanted to look at the effect of physical agents. And this, I think, is going to be interesting to you in an indirect way, and that's why I included it. This is the reactor that I was licensed by the Atomic Energy Commission to run while I was an undergraduate at the University of Wisconsin. North Carolina State also has a reactor. I think it's still functional. Wisconsin still is. This is Bremsstrahlung. It causes that beautiful blue glow. It's beautiful. Anyway, that was my undergraduate career. So electromagnetic spectrum goes all the way from radio waves up to cosmic rays, through visible, infrared, ultraviolet, all that kind of stuff, into the high energy gamma rays that are used for radiation therapy, to the diagnostic radiology energies. And the studies that I did were in this range of energies. The diagnostic range of energies are what we use for these low doses of radiation, their effects potentially on the epigenome, using the Agouti mouse model. So this is sort of radiation biology 101, but it's important because it'll come up why we did these studies. So you can affect, radiation can damage DNA and other proteins too, but DNA either directly by knocking out, let's say, an electron, which then breaks the backbone of the DNA or causes a mutation, or the photon can come in and directly damage it. There are only 2 ways in which you can protect yourself from this type of damage. Does anybody know them? Any idea how you protect against radiation damage? You.
Don't get radiation?
That's one.
Put some lead around you or something?
Lead. Lead is one. Does anybody know another way? There's one more way that you can protect yourself against it. Run. Distance. Only 2 ways. But that's only 20% of the damage. 80% is through the generation of reactive oxygen species, right? That can be protected from using antioxidants. So you have the potential of protecting against 80% of the damage due to radiation by use of antioxidants. We'll get to that later. So this study was a little bit more difficult. It only ruined the careers of 3 postdocs and graduate students, but finally one persisted, and that was Autumn Bernal, the last graduate student I had. So it was difficult because we couldn't just radiate like we did with chemicals. We put the mothers on this throughout the whole thing, and we didn't know exactly where it happened, but there was enough there that you could see an effect. Here we had to radiate. The nice thing about radiation, you know exactly when you irradiate and you know the dose. But the problem with that is that you have to irradiate now always at the same time. So these were all time pregnancies for animals, and which made this study a lot more difficult. Anyway, Autumn was able to do that. We irradiated 4.5 days after fertilization, and I'm now going to tell you the results of what we found. So I'm only going to show brown animals and yellow animals. So those are the two extremes. Remember I said in the controls it was about 10% yellow and 10% brown, and the vast majority of them were up 80% was here in the middle. And the doses that we used are, so you can get an idea what we were looking at. 0.1 centigrade is what you would get if you got a chest x-ray, for example. Or in the old days, that would be 0.1 rem or 0.1 rad. And a CT scan is about a centigrade. So in other words, if you CT scan your whole body would have 1 centigrade of exposure to radiation. To put that in some perspective, legally, if you work in a radiation facility, you can get 5 of these throughout the year, legally. So those are the doses we're using, very low, not therapeutic doses, these are diagnostic doses. The other thing I want to say is, and we'll get to this later too, is there's a thing called a linear no threshold model for radiation risk. So what that means is if you subtract background off, that the curve for risk goes through 0, 0, and then goes up linearly. What this means to you is that it says that there is no dose of radiation that's safe. They're all bad. Okay? So what do we think? If you were to set up this thing, what would you expect? Yellow is bad, brown is good. In a dose response manner, what would you expect to get? Yellow. That's what we expected. And I was like getting no information. This is bad. When your graduate student doesn't talk to you. Right? Like I'm saying, finally, Autumn, what's going on? She said, well, it's really freaky. And that's not a good word either. So this is what we had. Yellow. Dose response. Concomitant with this, now just think of this. Is this not freaky? She came in and she said freaky. What she said is there are absolutely no yellow offspring. All the offspring are brown. This is not at a level of radiation that selects because we had the same incidence like you were talking. We didn't see that. Same incidence of offspring, I don't know, whatever, 6, 7, 8 pups per mother. No change in that whatsoever. You would run into trouble if you got up to a dose higher than this, which we didn't go up, which I wish we would have still pushed it. But we didn't go up because we thought the direct effects of radiation would start actually killing the offspring so that you couldn't tell whether they were yellow or brown because they wouldn't live. But that's not the case here. So what low doses of radiation did was take that dose, that nice little normal distribution, and shoved it over to brown. So these are the results. We then looked at methylation. As you would expect, if this was indeed correct, you should see hypermethylation, and indeed we see it going up. But interestingly and very importantly, you start seeing it come back down. So it gets to a maximum of positive adaptation and then starts losing it. If we would have doubled it and the animals would have been alive and we had no problem with selection, my guess is that you would go back to almost the control value. But we didn't do that. But these are the results that we can say low doses of ionizing radiation increase the incidence of brown offspring by enhancing DNA methylation. I think that's pretty clear. Thus, low doses of X-rays induce a positive adaptive response in these mice by altering the epigenome. Positive, not negative. So then I said, okay, if this is true, then if we feed the mother antioxidants, knowing that 80% of the damage is due to the generation of reactive oxygen species, we should see this go back to control levels. Otherwise, there's something nutty here that I don't understand at all. And this is the experiment then that Autumn completed with. So if you look at controls, the ratio, as I said, there's about 10% brown, 10% yellow, the ratio is 1. So look at the little circle. Then at 3, that's the maximum effect we see. The ratio of brown to yellow is almost 15. 15-fold increase in brown animals compared to yellow when you radiate the mothers and the offspring at 4.5 to 5 days after fertilization, which means you're hitting the embryonic stem cells at that point. And then when we add the antioxidants, we go right back to control. If you look at methylation, that's control, average, because there's, about 50-50, that's the average level, about 20-25%. You at the optimal for brown animals, you have a very significant increase in methylation, which you would expect because there are many more brown animals or nearly brown. You add antioxidants, you go right back down to control. So the antioxidants negate the increased DNA methylation and block the positive adaptive response induced by low doses of X-rays. So now you've got something that people think are absolutely devastating, causing positive adaptive responses, and things that people think are wonderful, which are antioxidants like vitamin C and vitamin E, being absolutely devastating. I said, Autumn, we're never going to get this paper published. It ain't going to happen. It did get published. And the next thing is then the positive adaptive responses induced by low doses of X-rays is mediated in part through oxidative stress. So it doesn't, I don't think, make much difference how you stress the system. You're going to get these phenomena occurring. So now I want to finish this off with this last slide. And I like this because Rankin discovered X-rays in 1896, and he very soon took an X-ray. Notice that he took an X-ray of his wife, not himself. That's bad language. Yeah, you could tell by the ring, right? So this is what I've been talking about, the linear no threshold dose response model. So in this case, we have yellow obese mice, which are bad. These are negative effects. And the model system basically has been generated through doses that are very high in the therapeutic level, and then they're extrapolated down. But what we saw was something that looks more like this. And I want to go through this because I actually forget every time to make this clear. So when you're here, in other words, when radiation is causing a positive adaptive response and you add antioxidants, you move back up to control. That's what you're doing. The question that I wish the hell we would have done, but we didn't, and it needs to be done, what happens if you were here, just at the level of where things are toxic, and you now add antioxidants? My guess is that what you would do then is move it back down to here, and antioxidants would now look like they're advantageous. So the effect of antioxidants on this system kind of depends on where that system is when you give the antioxidants. And that's why you see, I think, studies with things all over the darn place. They don't replicate, it doesn't make any sense. Because you're moving things up and down this curve and you don't know where they are when it's being given. Only a possibility. You guys need to do these things, and you might do these studies. I mean, these should be doable in your animal systems with temperature. Now I'm going to introduce the one time I say temperature. It's possible that increased temperature could be doing the same types of things. I don't know, but it's something you guys now, I think, are looking at. So this model system is not compatible with the linear no threshold model. It's not even compatible with the threshold. There is not a threshold. There actually is a positive adaptive response. This is compatible with the hormesis model of dose response. And there's now talk in the United States anyway about potentially replacing this linear no threshold model with something that has a little bit more biological relevance. So questions to end. Do other frequencies of electromagnetic radiation or even high energy particles also alter the epigenome? We don't know. This is important because we're planning to send humans in deep space where they're going to be irradiated. This is the most important probably. What is the signal transduction pathways or pathways that link the formation, it appears, of reactive oxygen species to altered DNA methylation? I don't know, but I think this has to be looked at biochemically because this sensing system seems to be the redox state of the cell, and that's linked to the machinery that's laying down these programs. But what are the wires that go between these two? I don't know what they are, but they need to be identified. Does low-dose radiation alter the human epigenome? We don't know that either. And is the linear no threshold potentially inappropriate risk assessment model? My guess is that indeed it is. And now one could put, does low-dose temperature or high-dose temperature affect this too? We don't know that either. So in summary, we have now shown that exposure of offspring to both physical and chemical agents while in the womb alters adult disease susceptibility by modifying the epigenome. It's incredible to be able to actually take all of the work that we've done for the last 20 years and bring it down to a single sentence. But the ability to say this with certainty really has changed the way we view disease susceptibility really forever. It's now altered. So I like to end here. This is the last one. Food for thought. There's two quotes, one from a physicist and one from a philosopher. And I want to introduce the physicist to you without giving his name because he was at the University of Wisconsin. He started medical physics. If any of your relatives have ever had determination of bone density, that system was developed by him. He developed the first system, solid-state system for detecting ionizing radiation. This was a brilliant physicist. Now, some people, after you read this sentence, this next sentence that he wrote just before he died, might think he's not a very good biologist. But this is what he said. I am now almost certain that we need more radiation for better health. Now, you can imagine how well that went over in the literature. But from the philosopher, which you all know, what does not kill me makes me stronger. And that's the end. Thank you.